Hello everybody. So today in the online course we'll be studying about a special topic which is called apoptosis. It got another name called the programmed cell death. So first of all, what actually this apoptosis is a systematic way of killing cells, or or you can say it's a programmed way of killing cells. So for a definition, it can be said as it's an orchestrated sequence of events that lead to death of the cell, or you can say it as the biochemical events which lead to the cell change into death. So now, why we need to kill the cell? Why we need to go for the death of the cell? So the why you need to go for this apoptosis? So let me tell you that every day the animal, they used to go they suffer from a lot of viral infection, or they go for they come under radiation, they come under chemical carcinogens, a lot of things. So under this circumstances, there are a lot of cells which got damaged. Their genomes may got damaged, their cytomembrane may got damaged. Under these circumstances, we cannot continue this damaged cell to go for cell division, or else it may transform into a cancer. So first of all, this apoptosis was it was coined by John Kerr, Andrew Willey, and Air Curie in the year 1972. So it is said that in human body, almost about 10 to the power 10 to 10 to the power 11 cells, they should die every day by apoptosis. As I have told you that why we need to eliminate cells for apoptosis. See, I've told you that it is important to avoid unwanted growth of the cell which may lead to the cancer. Suppose generally in cells, we know that if there's a damage in the DNA, or minor damage in the DNA, they got repaired by DNA repair mechanism system. There are a lot of different repair mechanism systems. So now if they suppose they cannot go for repair mechanisms, it is irreparable. Under such circumstances, they have to go for self-destruction of the cell, or else this damaged DNA, if they got divide, it may lead to cancer. And this is how actually the cancer they develop. That is, if there's a damaged DNA, they got divide, and thus they cannot control the dividing of the cells. It's uncontrolled growth of the cells which lead to the formation of tumor. Now this is apart from eliminating this unwanted cell from the body, this apoptosis got another function. It is during the developmental phases of human or other multicellular organism, they need to kill certain type of cells to form the organs. Let me give you this example. You can see this one diagram over here, that this cell mass which is developed during the embryogenesis. Now to form the finger, they have to kill these cells in between so that they can form the finger. Or in case of a frog, during metamorphosis, they have to kill the cells from the tail so that they can form an adult frog. So this is how during embryogenesis, even in development of brain, in in embryogenesis genetic phase, it got a lot of neurons in our brain. So by the time a baby is born, a lot of neurons, they got killed by themselves or by apoptosis. Or suppose this process also continued in the elder ages, which may lead to the diseases like Alzheimer's diseases, Parkinson's. So these diseases are neurogenic diseases. That means this disease, they destructs the neurogenic cells like hippocampus cell, which may lead to Alzheimer's disease. So what actually the apoptosis is? This is the brief outline of the apoptosis. So you can see a healthy cell. Suppose this is a healthy cell, but under certain some circumstances their DNA got damaged or some other factor, this is it become unhealthy. Now it cannot be divided, we cannot go for division, or else it may lead to cancer. So then it go for apoptosis. How? First of all, they will shrink in size, the overall volume of the cell will be shrinking, then the cytoskeleton will be broken, their nucleus will be shrinked, then their mitochondria will be shrinked, and finally they'll form a kind of irregular blebs. And when they form these irregular blebs, then they will emit some signal to eat them up by phagocytes. So these are the phagocytes which will engulf them. So basically healthy cell, they go transform themselves into blebs so that they can be eaten up by macrophages or the phagocytes. So this is the overall cycle of apoptosis. Now we have to learn that how they got to know that they need to be go for apoptosis. This is the molecular mechanism, or how a cell go for this configurational changes like shrinking of nucleus, breaking of this chromosome, breaking of cytoskeletons, how they go for it. So let's begin. So let's discuss about the history of this apoptosis. As always, the C. elegans or the Cinerobacteris elegans, these are key model organisms to study all sort of scientific studies. So the first study was conducted in C. elegans only. It was found that during a development, out of 1090 cells during development, almost 131 cells they die. That means what? This 131 number of cells they are getting killed, they are getting distracted or lost. So this indicates, yes, the cells they die during process of development. Then later in the 1986, scientists found that there's one gene called CED3. Suppose if this CED3 become mutated or it become dysfunctional, in this case what they found that there's no apoptosis take place in C. elegans. That means what? This CED3 is now responsible for apoptosis. That means if there's mutation in CED3, it is not functional, that means they won't go for apoptosis, which clearly indicates that the CED3 is responsible for apoptosis. The later one, the scientists carried on their research and they found a homologous set of protein is also found in mammal, which is produced from a set of gene called the caspases. This there are a lot of caspases, caspase 3, caspase 8, caspase 9, and they are all responsible for the apoptosis. In the right hand side you can see a molecular three-dimensional structure of protein caspase 9. Okay, next let's discuss that how this apoptosis is triggered, that is how they go for the signaling. And this can be done by two methods, either by an external factor or an internal factor. Let me tell you clearly, this external factor means those factors which comes from the outside the cells, like suppose a cell is under radiation. Suppose there's harmful radiations like gamma ray or other strong radiations, or certain strong chemicals, or like cytokines which is produced from the immune cells. Under these circumstances, the signals which comes from the outside may trigger the apoptosis in the cell, and they follow a pathway called the extrinsic pathway. And the other way, there's another way called intrinsic pathway, which is being stimulated by the internal factors, internal stimulus. That means the signal which comes from the within the cell. Like for example, if there's a damage in the DNA, or you can simply say the irreparable genomic damage, or in case of hypoxia, the lack of oxygen in the cell for a prolonged time, or high calcium content in the cell, or you can say a viral infection in the cell. Under such circumstances, the cell they bring out their own signals within the cell. From then they go for a pathway called the intrinsic pathway for the apoptosis. So we'll explain one by one both extrinsic pathway, how they follow this extrinsic pathway, and how they go for this intrinsic pathway. So first let's discuss about this extrinsic pathway. That means if a signal which comes from the outside the cell, like a radiation, a chemical, or a cytokines, etc. So they follow the extrinsic pathway. Now the beginning of this pathway is start from the TNF, or simply tumor necrosis factor. Now why they are called the tumor necrosis factor? Because they got the ability to kill or go for necrosis of tumor cells. They can kill the tumor cells. So let me tell you, the doctor who go for the combating of cancer, they used to treat the patient with radiation therapy or the chemotherapy. Now this chemotherapy or the radiation therapy, these are the external form of the stimulus, the extrinsic stimulus. Then they trigger the cells to go for apoptosis, self-destruction, so that the cancer cell can be killed, and this is how the cancer is combated, the cancer is cured. So this TNF, which comes in the form of a necrosis factor, which come in the form of radiation, or it can come as a chemical, or it can come as a cytokines. So when they reach the cell, so first of all, outside the cell they got a receptor particularly specific for this TNF, that is tumor necrosis factor, and this receptor is called TNFR1, or tumor necrosis factor receptor 1. Now this TNFR1 is associated with certain protein which are called the death domain protein. For example, FADD is also known as first associated death domain protein, or TRADD, which is also known as TNFR1 receptor associated death domain protein, and a special class of protein which is called the procaspase 8. So this is one unit, a complex. This whole together, this TNFR1 is there, this FADD, TRADD, procaspase, they all form one unit of TNF receptors. The moment when this TNF reaches the cell, they reach the TNFR1, there will be a configurational change. Where? In procaspase 8. Here, this is the protein. Now basically this procaspase 8 is inactive. It is named as a procaspase. Whenever there is a pro, that means it is always inactive. So procaspase 8 is inactive. The moment TNF binds here, they go for configuration change. They cleave out certain unnecessary protein which makes them inactive, and thus when it got cleaved, they become initiated caspase, or it becomes active. Now they do not have a direct role in apoptosis. Our ultimate aim is to make this executioner procaspase into executioner caspase. Now this executioner procaspase is available in the cell, inside the cell, but they are inactive. Executioner procaspase, this pro is there, that means what? It is inactive. This executioner procaspase, it is inactive. So our aim is to make them active. Who will make them active? This initiated caspase will make them active. How? Cleaving out the same way, the cleaving out the unnecessary protein which makes them inactive. So after cleaving out this protein, basically they are the proteolytic protein. So they cleave out this protein and they become executioner caspase, or the functional one, and now they go for apoptosis. So I'll explain again. This is one complex present in the cell which is known as TNFR1, or tumor necrosis factor receptor 1, which is associated with lot of death domain along with procaspase 8. The moment the extrinsic factors stimulus comes from the outside and reach the cell, there will be a configurational change in this protospase 8 which will make them into active initiator caspase, and once this initiator caspase is activated, they will make inactive executioner procaspase into an active executioner caspase, so that now they can go for apoptosis. In the end of the chapter, I will explain how this executioner caspase is they go for apoptosis, the cellular process. This is the mechanism of molecular mechanism of transaction of the signals, that from how this external stimulus they go for apoptosis. So this is extrinsic pathway. That means whenever the stimulus is from outside, then how they transfer the signals to go for apoptosis. Next is the intrinsic pathway, when the stimulus comes within the cell, from the cell. This can be due to the damaged DNA, viral infection, high calcium concentration, etc. Now, this intrinsic pathway, they are regulated by a special class of protein called the BCL2 family protein. Now, this BCL2 family protein, they are subdivided into three groups. Number one, the pro-apoptic protein, that means which promotes apoptosis. There is another class of protein called anti-apoptic protein, and there is another class called the regulator protein. So first one, the pro-apoptic protein. Example of such protein is the Bex and the back. That means this class of protein, they are hungry for apoptosis. They are ready for apoptosis. They want to go for apoptosis. But this is totally antagonistic, anti-apoptic. They do not want to go for apoptosis. They restrict cells to go for apoptosis. They initiate cells for apoptosis, but they restrict cell for apoptosis. Now this one is the decider, the BH3, that promotes apoptosis indirectly. They are the regulator. So these are the different class of pro-apoptic, anti-apoptic protein available in the body. So now this class of protein is often asked in net questions, that these bags and bags are anti-apoptic or pro-apoptic. The same with the BCL-W, BCL-XL, they are anti-apoptic or the pro-apoptic. So such questions you can expect in CSR life science net examinations. Now, one important factor is that how this BH3, they decide whether they have to go for apoptosis or they have to restrict apoptosis, or suppose they need to go for apoptosis, how they go for apoptosis. The next slide, I'll show you that BH3 promotes apoptosis by two ways. Either by inhibiting the anti-apoptic protein. That means this class of protein, this anti-apoptic protein, that is BCL-XL and BCL-W, they are against the apoptosis. They do not want to go for apoptosis. So BH3, what they do, they is to inhibit them. They have to suppress them to go for apoptosis. The second way of activating is that you have to activate the pro-apoptic protein. They are the protein bags in the bag. They are the hungry protein which are hungry for the apoptosis. They always wanted to go for apoptosis. Now it is in the hand of BH3 that how they will go for apoptosis, either by inhibiting anti-apoptic protein or by activating pro-apoptic protein. Either way, these are the two ways they go for apoptosis. Now, how they go for apoptosis? This is a very crucial mechanism, and is somehow different from the extrinsic pathway. The extrinsic pathway, it is kind of direct mechanism, but here it is bit indirect. So the moment the BH3 they allow this pro-apoptic protein like Bex and back to go for intrinsic pathway of apoptosis, what they do, from cytosol of the cell, they migrate into the mitochondrial membrane. The the mitochondrial membrane is got two layers, outer membrane and then inner membrane, and in between them they have perimitochondrial space. So this bags and back they get inside this mitochondrial membrane, and what happens after that is that the mitochondrial membrane, they got a change in their permeability. Got my point? That once the bags and the back protein they got inside the mitochondria, the mitochondrial membrane, there is a change in the permeability, which in turn allow the cytochrome C is a mitochondrial protein which is present in between the perimitochondrial space. I have told you the space between the outer mitochondrial membrane and the inner mitochondrial membrane, there is a space called the perimitochondrial space. This cytochrome C, they were present over the perimitochondrial space. Now since the permeability has changed because of insertion of bags and the back protein, the cytochrome C, this starts coming out of the cell, out of mitochondria. Now when cytochrome C start coming out of mitochondria, what they do, they got attached with one protein called FF1, and ultimately they got linked with the procaspase 9, and finally they form a unit called apoptosis. There are 1 2 3 4 5 6 7 7 unit of FF and procaspase 9 and cytochrome C. So you can see these green portions, they are the procaspase. This is the illustration, not the exact diagram. This is the illustration how they work. So for example, this is procaspase 9 and this is FF, and they got active only when the cytochrome C is present. So this apoptosis is inactive, they remain there as a procaspase 9, and FF1 is always available in the cell, but they are inactive. Why? Because of the lack of cytochrome C. And how cytochrome C comes? Once the bags and the back protein they got inside the mitochondria, then the cytochrome C will release from the mitochondrial and then it come out, and then they link up with the apoptosis to make them active. And what is the function of this apoptosis now? Again, the same way, the way in extrinsic pathway, our ultimate aim was to activate the executional procaspase into activated executional caspases. The same way here also, this apoptosis, it only function is to make this executional procaspase which is present in abundant in the cell but in inactive form. So this one, it will make it active. How it will make active? By cleaving out this unnecessary protein, and then it will become executional caspases. And then the way in extrinsic pathway, in intrinsic pathway as also they will follow the same apoptosis method. Got my point? I'll explain again. The moment BH3 family protein allow the bags and the back protein to go for intrinsic pathway, so this bags and back protein, they will get inside the mitochondria, and once this mitochondria is get inside the once this bags and back protein get inside the mitochondria, there will be a configurational change in the permeability of the mitochondrial membrane, which in turn allow the cytochrome C present in the mitochondria to migrate out of the mitochondria, and it will link up with the apoptosis, and thus apoptosis become active once they got associated with the cytochrome C. And once this apoptosis is active, they will cleave the executional procaspase, and it will make it active executional caspase. And when this executional caspase is active, they can go for apoptosis. Now how in both the way, extrinsic pathway, in the intrinsic pathway, our in both the pathway the executional caspase was activated, isn't it? So now how this executional caspases they go for apoptosis? They basically they target certain protein in the cell and some vital protein which are necessary for survival of the cells. For example, they First of all, they target all the protein kinases and focal adhesion kinases, protein kinase B, protein kinase C. That means what? These are this protein kinases. I guess you all know that the kinases, they are important in activating a lot of enzymes and the hormones. And the especially this focal adhesive kinase is responsible for the addition of cell to cell. For the joining of two cells, they need this FAK. So once this group of protein is being targeted by the caspases, targeted means they got distracted, and once this protein is missed, that means what? It will lead to the detachment of the cell from the neighboring cells. So it will this broken it will be loosen up from the normal cell mass. Then the next target of the protein is the lamins. Now you know that lamins, they are present inside the nuclear membrane. Now if lamins got broken, that means there will be disassembly of nuclear lamina or nuclear membrane, or the overall nuclear structure will be damaged. Then they target all the cytoskeletal protein, microfilament, intermediate filament, microtubules. They got damaged by this caspases, and so once they got damaged, that means this cytoskeletons, they are very much important in maintaining the shape of the cell, size of the cell, and maintaining the cellular organelles in the position. So that means once the cytoskeletal proteins are got damaged, that means there will be change in the shape of the cell, everything will be damaged. And there's one more important protein they target, that is called the caspase activated DNase. That means they activate one DNase. DNase means those are the enzymes which eats up the DNA, which breaks the DNA. So that means what? This CAD or caspase activated DNase, it will attack the DNA and it will break the DNA into fragments. Simply you can say the chromosomes, all the chromosomes, they will be fragmented into pieces. And once these things happen, that means once the cell is detached from the cell mass, normal cell mass, once its nuclear membrane is damaged, once its cell shape is changed, it will broken into blebs, DNA got fragmented, then ultimately what? As I have told you here, yes, these things happen, right? There is a shrinkage of cells done by the those protein damage in the protein. Ultimately it will be engulfed by the phagocytes. So after this process, the cell, when it will become individual, it will become damaged, then it will be engulfed by the phagocytes, and thus the apoptosis process is complete. So thank you very much, and please stay safe.